Our next speaker will be a team from University of Washington, consisting of Rachel Wong, Fred Ricci, and Thomas Ray, who will be giving the talk for the team.
Well, thanks. And thanks very much to the Allen Foundation and to Paul Allen for his vision in really setting up these groups for innovation. It's kind of amazing to me what one person can do, the kind of impact he had, just funding all this kind of research that's going on here, but also this Brain Institute. So it's pretty amazing. Anyway, what I'd like to do today is talk about, kind of give a wrap up, I guess you could say, of our project that we've been doing over the last 3 years. And we got interested in this whole concept of trying to understand circuit development in both normal development of the nervous system and potentially in organoids. And so when we wrote this grant to apply for this funding, what we really hoped to accomplish was obviously way more than what we ultimately accomplished. But I think it doesn't hurt to be ambitious in this kind of a context, and so we were. And basically, our goal was to try and create a mature neural circuit entirely in vitro from embryonic stem cells. And the neural retina was the place that we chose, both because the three of us had been studying the retina for quite some time, most of our careers, and we realized that in terms of organoid development, the retina was perhaps further along than some other areas of the nervous system. And so that we thought we would have some chance at actually getting the mature neural circuit to develop. I'm not going to be able to tell you today about all the different things that we did or all the different projects that were going on, but I'm going to highlight two of them. The first is really well, I'll get to it in a second, but basically the other, this was the other rationale that I've described. The other thing is that there was already a wealth of knowledge in the retina about its circuitry. Now, the retina is a laminated structure, just like the cerebral cortex, and in some ways this laminated structure reoccurs in organoids. So that was really something that I think has been known since some of the really pioneering work on organoids by Yoshiki Sasai. And so these basic cell types, the retinal ganglion cells here that project through the optic nerve, the inner retina, bipolars and amacrine cells and horizontal cells that occupy this layer, and then the photoreceptors, the rods and the cones, occupy that layer. And so, but when we started this project, we already knew that we could generate these various cell types and that some lamination occurred, so that gave us hope that we could ultimately understand the circuitry development. The circuitry of the retina is built up over time developmentally in sort of waves, where initially you get a set of early cell types, ganglion cells, horizontal cells, and cone photoreceptors. And then later, the rod photoreceptors begin to appear, and amacrine cells. And then finally, bipolar cells and Mueller glia, and further development of the rods and the cones. And so this sequence is not only a sequence of generation of each one of these different cell types that's similar to the cerebral cortex building itself up by layers, but also in terms of their maturation and circuit development. But what was not really known at that time, though, just a few years ago, and so what we really needed to do, we set this up in parallel where we were going to be studying mouse RNA, or mouse development and human development, both human acutely isolated retinas from fetal stages and human in vitro, and then finally human ES cells. And we decided to focus on the human, partly because there was already some detail known about the mouse, but ultimately, what we want to understand is how the human retina develops and potentially create models of that for human disease. And had the Allen Brain Institute, been the Allen Brain and Eye Institute, we wouldn't have had to do much of what we had to do. But what we had to do was actually really characterize the development of the human retina, because there really was very little known about it. And so really our first project was to obtain human specimens from a variety of different ages of fetal development, from 52 days to 136 days. We did bulk RNA-seq analysis of these things. This is a 59-day human retina, 96-day human retina. We can go all the way up to 140-day human retinas. And we did extensive characterization of the cell types, the cell type appearance, the normal circuit development, correlated that with RNA-seq at both the bulk-seq, as you see here, which was published last year, these data in developmental cell, but then more recently we've extended this to single-cell RNA-seq and done the kind of analysis that I think will really fully characterize human fetal development through this range of stages. So we sort of created our own atlas. In addition, Fred Ricci began to record from these cells, and Rachel then developed a kind of a mini connectomic project for the various stages of the connectome. Now, I'm going to tell you a little bit about what Rachel found, which her lab group found. I'm really not going to have time to go into what Fred really has been working on. But one of the things that we focused on was the thing that makes the human unique. And it's the thing that you're really using right now to look at these slides. And that's the fovea. The fovea is a highly concentrated zone of cone photoreceptors that the primates focus, use to really see the world in great detail. Now, the circuitry development, we already knew some things about how circuits develop between mouse and human. And we knew that they were roughly the same. Initially, the cells are generated, then the inner plexiform layer begins to form, and then the outer plexiform layer begins to form. And these are the synaptic layers that allow the circuit, the connectivity. But what we didn't know, because it really hadn't ever been studied before, was how the foveal circuit develops. And that's partly because mice don't have a fovea. And so this has to be studied either in humans or in non-human primates. So the fovea is a very unique, almost single channel of information that comes from a single cone to an on or an off bipolar cell, to a midget ganglion cell, a pair of on and off midget ganglion cells, and then into the brain. And this unit, this fundamental unit of your foveal vision is repeated then 1,000 or more, 1,000,000 times, so that we have this high density foveal vision that begins our appreciation of the detail in the world. So how does this develop? Well, we really didn't know. But Rachel undertook a serial block face electron microscopic project where they section through the fovea of developing fetal human retinas at various stages. And I'll present a little bit of that data today. But basically, the take-home message, there were two hypotheses. Either it develops with this kind of specificity right from the get-go, where cells selectively target individual cells that they're ultimately going to be partners with, or does it develop in this way where you have multiple inputs and multiple outputs, and then you ultimately select among those. Now, this is the kind of thing that we think of in terms of plasticity and visual cortex and in some of the higher order brain centers where function then refines the connectivity. But this is really more typical of what happens in the mouse retina. And so the question was, what's it going to be? So she began these electron microscopic EM serial block face reconstruction studies, and we were able to reconstruct bipolar cells. These are the cones, these are the bipolars, and really create a bit of a detailed connectome of the fovea at several different stages of development. And then also do the same kind of connectomics for the inner retina. And so we could look at the number of partners, the number of synaptic ribbons between a given bipolar cell, and how many ganglion cells does it synapse with, and conversely, how many ganglion cells then does each bipolar project to, and how many bipolars project to a single ganglion cell. And through this kind of analysis, what she actually found was that it looks like synaptic refinement is going to be necessary for foveal vision to shape the midget circuit. That both ganglion cells and bipolars initially have this model, where you have a couple ganglion cells connected with a given bipolar cell and several cones connecting to a given bipolar cell. And over time, it becomes more refined with really, first, we see it we see the on layer of the ganglion cells develop, and then the off layer cells develop until they finally have this mature circuit. And so we're continuing to build on these studies. We're getting ready to submit this work, but there's such a rich detail of complexity that comes from the serial block face that we continue to now look at how some of the other cell types are integrated into these circuits, the amacrine cells and the horizontal cells. But in the fovea, this is really uncharted territory, and so it's been really fun to assess how this circuit is developing. Now, this is the circuit developing in vivo, and most of our RNA-seq studies were done in vivo. But we've also been doing parallel studies in organoids. And what I'm going to tell you about next isn't really the circuit development in the organoid, but one of the motivating features that got us really interested in this in the first place was to try and understand a little bit more about what regulates developmental timing. Because all of these events I've described to you all occur with a regular sequence in development, and they also occur with that same sequence in organoids. So mouse organoids develop on mouse time, and so a 3-week-old mouse organoid is equivalent to a newborn mouse retina, and likewise a human retina will take 3 months to get to that same stage. So what we asked the question, what is this species-specific developmental time? So mouse and human, outside the fovea, both follow a very similar sequence of development. We've done parallel studies on human RNA-seq, as I told you, and mouse RNA-seq, and they follow very similar patterns of development. In fact, it's approximately 95% of the genes in mouse and human retinas follow the same pattern if you look outside the fovea. So the difference is mice do this in 2 weeks and humans do this in 12 weeks. And quite remarkably, human organoids develop with the same time course. So a day 40 human organoid has the ganglion cells forming just like this case, but we have to wait to 70 to 110 days to start getting rods to develop and get to this stage of the circuit. So the human time, and part of our motivation for this grant was how do we speed up human time to be more like mouse time. Now, if you think about time and what regulates time, time is related to organism size in a way. Developmental time has to do, we're slower than mice because we're a lot bigger than mice. We have to get a lot more cells than mice. And in general, for mammalian species, time of development scales, gestational age scales with size. But what's quite interesting is organoids, this is a human fetal retina at 59 days. This is a organoid at about the same stage. We're getting all the same cell types. We're getting the ganglion cells. We're getting the developing photoreceptors. This organoid is approximately this point of the human fetal retina in terms of its ratios of cell types. But the organoid is a lot smaller. It's gone through way fewer cell divisions, but it's right on time in terms of the development of its cell types. And we've looked at that with RNA-seq as well. We've looked at it with single-cell RNA-seq. We've looked at it with these sections. So organoids, human organoids really develop on time. It's not the overall size. It's not the number of cell divisions. And it's a similar rate in isolated tissues then, because the organoids were really generated from cells, and they didn't go through they went through a stage where they were really directed to develop retina and eye tissues, not making all the tissues in the body and then isolated. And so that's really, the fundamental question I want to address in the next few slides. So one of the earlier meetings here, I met Steve Horvath, and Steve told me, and Steve showed his data on the methylation clock, DNA methylation clock of human tissues, and how this worked across all human tissues. And as Steve points out, this is one methylation clock. There's many methylation clocks, but it's 353 CpG sites that cross the genome that correlate with age. And just to take a look at this, these were samples from cerebral cortex across the lifespan, from 0 to over 80, and they fall nicely on a line that Steve has called the DNA methyl age, and it correlates very well with chronic. So we wondered if we could extend this back to fetal samples, and we sent him fetal samples, and sure enough, they follow the same clock. Obviously, the scale, things are happening much more quickly, and I need to go much more quickly in my talk. So the next few slides I'll go through pretty quickly. So human fetal retinas continue to have normal developmental time even when we culture them, and this just to show you they progress normally, and they also progress in vitro with the aging clock. So the blue dots show the cultured ones, and the gray ones show the uncultured ones, and these were pairs. And so the aging clock ticks normally in vitro and in vitro, and we can follow DNA age in this way. So we thought that might give us some insight now that we have a clock of DNA methyl age that we can follow along with developmental time, and that maybe we could understand both processes a little bit better. But we did notice that even though foveal development is much further ahead, that its DNA methyl clock falls right with the periphery. So that was a bit puzzling because this was a disconnect between DNA methyl age and developmental time. And we also found that retinal organoids, although they nicely follow the clock and they follow the same developmental clock, as I said, they're a little accelerated. So they should be down here, and instead they're up by about 6 months further ahead. So a second disconnect. And then lastly, we looked at people with trisomy 21 because Steve had previously shown that accelerated epigenetic aging occurs in Down syndrome by a number of measures. And so we took fetal samples that were Down syndrome. And these 4 blue samples are the Down syndrome fetal samples. And they all show accelerated aging even in fetal tissue. So Down syndrome people get a lot of age-related diseases early onset in their 40s. And I think that's what motivated Steve to look at this accelerated aging clock. But we already see this signature of accelerated aging in these fetal tissues of Down syndrome patients. So this suggests that the aging clock and the developmental clock might be independent because we don't see a big change in the RNA other than genes on chromosome 21 are increased in the Down syndrome retinas. Other than that, we don't see big changes in the RNA-seq or in the immunostain for the different cell types. So basically, this is kind of my summary slide of the DNA epigenetic clock and the developmental clock. We got into this thinking that the epigenetic aging clocks might provide an independent measure of developmental time so that we could better calibrate our studies on developmental time. But what we found is that sometimes these two clocks are in synchrony, and sometimes these two clocks are not in synchrony, but it's interesting. There's a lot of phenomena here that I think needs to have some better understanding epigenetically. So what we're doing now is we reasoned, well, maybe we can take the differences, the asynchronies in developmental and aging clocks to try and inform us as to the mechanisms underlying one or the other. So the genes that increase in the trisomy 21, but the genes that are the same across the different retinal regions where the developmental clock is different but the aging clock isn't, and then the genes that increase in organoids because they have an accelerated aging clock but a normal developmental clock. So we can sort of parse out, using these different categories, what's the intersection across those different conditions. And it really contains a small number of genes that may allow us to better test whether some of those genes are involved in the developmental clock or in the aging clock. And so those experiments are ongoing, but I think will be a unique way to approach this question of time. So with that, I think I'm out of time. And so I'd just like to thank Fred and Rachel, Akina, Chizang, and Akshaya. And again, thanks to the Frontiers Group, and thanks to Paul Allen.
We have time for one question. I'm sorry.
Yes.
So Tom, obviously whatever encodes developmental time has to be decoded in the temporal series, right?
Yes.
The fact that you generate different cell types at different time points, and that is consistent. And that would have been one of the early signs that the methylation clock would not necessarily, because it's very hard to see how that would be decoded by these temporal fate series, So what is there any hints in the genes you say overlap between those three Venn diagrams that would suggest how you might link sort of total, real physical time to to be decoded
Right.
by this sort of sequential cell fate.
Right. Well, one of the things that we had found earlier that stops the developmental clock was a dicer knockout, and this is in mice. And so we thought that perhaps microRNAs were involved in that, and for that reason. And so we actually found that LET7 and microRNA 125, miR125, were important in regulating that developmental clock in mice. Now, interestingly, those genes are on chromosome 21, the genes underlying those microRNAs plus miR9, and those are upregulated in this case. But that's the developmental clock that we found was regulated by that, this sequence of cell types generation, not the aging clock. And in fact, Down syndrome is the opposite. Down syndrome gives you an accelerated aging clock, but not a big change in the developmental clock. So it could be something like that. The genes that we did find, a couple of them had been involved, though they're not the dominant ones in the aging field that we think that were in that intersection, but they have been implicated in aging syndromes, in mutations and SNPs and things like that in the past. So whether or not those would allow us to understand how these two processes are coupled or not coupled, yeah, I'm giving you a hand-wavy answer because I really don't know. I mean, I think these are just more exploratory phenomena at this point. We're observing, we're wondering. If you have good ideas, please let me know. Thanks.
Okay, thank you so much.